Well, thanks so much for the invitation to speak with you this afternoon. I really appreciate it. I've, I'm very familiar with some of the work that your research center has done, perhaps not all of it, but looking into it a little bit, you've been able to mix chemistry and biology and biotechnology in a way that I think is extremely compelling. You've had so much success both in applied and fundamental science and dealing with practical problems that touch on varied sectors of the economy, including agriculture, waste treatment, production of fine chemicals to add value to feedstocks, issues related to environmental sustainability, and broadly the bioeconomy, and keeping in mind how these various aspects are applied to human health, whether it's concerning food safety or mitigating the development of antimicrobial resistance, which is an important challenge. And the vision of your center has been very international, notably as evidenced by your deep implication in the priorities of the European Union. So please accept from me my respect for what you've been able to accomplish over the last 30 years. My congratulations, and my hope that the next 30 years with the upcoming generation of young scientists will be as or more successful. So with that, I've been asked to spend a few minutes talking about human health within the context of the One Health Continuum. Can you see my next slide?
Yeah.
Okay. Thank you. So just to give you an idea where I'm coming from, if I'm coming from far away, if you look at the left-hand corner of this image, you'll see a map of the world. And if the laser pointer is working correctly, you are over here. I am over here. Here in the top right-hand corner, you see a map of North America. Within North America, there's the Great Lakes Basin at this point here. And within the Great Lakes Basin, you might be familiar with the city of Toronto, which is quite a large city. And I'm based in a city called London, Ontario, which is roughly 200 kilometers southwest of Toronto. I was looking at the weather that you're enjoying in Porto this week, and it's very lovely. Here's an image on the bottom left-hand corner of my front yard at the moment, under approximately 30 centimeters of snow. So it's very different from what you're living. So what I'm going to do over the next few minutes is talk about the One Health Continuum. Why should we be thinking about One Health? And talk about that in the context of antimicrobial resistance, food safety, and with a couple of concluding comments. The talk will be quite AMR centric because that's what I'm most familiar with, but hopefully touch on some other things that you hopefully are interested in. So One Health. So famously, Leonardo da Vinci is quoted as saying, learn how to see, realize that everything connects to everything else. And that's probably the simplest and the most profound way of thinking about this One Health idea, especially from the perspective of microbiology. So one of the most interesting, to my mind, developments in microbiology, at least in my career, is the deeper understanding that each individual, each eukaryotic individual, all the way from an invertebrate to a person or an elephant, is really an ecosystem which comprises the eukaryotic organism and a prokaryotic complement. And so humans and other primates and mammals have been on Earth for perhaps up to 100 million years, perhaps less than that. Whereas bacteria and archaea have been on the planet for perhaps 3 to 4 billion years. And so they have vastly more evolutionary experience than we do. And we eukaryotes have learned to exploit this for our benefit, for a variety of functions that are absolutely essential. For example, digestion of food, provision of essential nutrients that we cannot biosynthesize, appropriate priming of our immune system through interactions of our gut microbiome with the gut. And so that has been of extreme mutual benefit. But actually, that perspective is particularly human-centric and naive. We're really just chemostats for the prokaryotes. They are exploiting us. But anyway, we do have this profound interaction with the microbial world. And of course, there's now extensive interest in manipulating the human microbiome for the benefit of health. And actually in agriculture, we're trying to deploy some of those lessons to look at the microbiomes of animals as well. And so the microbiomes of people, of individuals, are interconnected as well. So at birth, a baby is seeded with the microbiome of her mother. There's some evidence that if that doesn't happen, it's to the detriment of the health of that individual. And also, if you look at the microbiomes of individuals within a household, they are closely linked as well. So that the idea here is that individuals are sharing their microbiomes. The microbiomes of humans and animals are connected as well. And so if you think of the animals with which you share your homes, they share your microbiomes as well. And that may be a good thing or possibly a bad thing. For example, there's a recent report that dogs are carriers of Clostridioides difficile, which is problematic for human health. Extending that idea more broadly, humans and food animals are connected. And this is very relevant to food safety. If the food that you're consuming, meat, for example, or dairy products is contaminated with organisms that are of human health concern, you can become ill. And the connection between humans and food animal microbiomes is not only evidenced through acute illness, but also probably through other kinds of organisms that have more subtle effects, if you will. And so there's some evidence that the gut microbiome can become seeded with E. coli that probably amongst other sources, but certainly poultry meat would appear to be a source of E. coli that under certain circumstances can cause urinary tract infections in humans. And food animals and people are connected to the microbiomes of wildlife. So in this particular example, you have avian influenza that's carried within reservoirs of wild fowl that can interact with domestic fowl under backyard or more commercial circumstances, and contaminate the fowl or the chickens or ducks with viruses that may be fatal for them, and in some instances may be problematic for human health as well. And finally, the distance through which these microbiomes are connected can extend from the local to the global. And in this particular example, this is a Dutch study where they looked at several thousand individuals who were traveling to countries outside the Netherlands. And they took fecal samples of these individuals prior to their trips and following their trips. And they looked for carriage of extended spectrum beta-lactamase Enterobacteriaceae before and after these trips. And if you look at based on a large data set at the percentage of travelers that acquired ESBL Enterobacteriaceae, in some instances it was quite high, and it varied according to where the individuals traveled to. So if the individuals traveled to the Indian subcontinent, roughly 3 out of 4 individuals brought back with them ESBL Enterobacteriaceae. If they traveled to China, roughly 1 out of 2. And as you can see, if they travel to other countries in Africa or in South America, the percentage acquisition that came back varied accordingly. So what do we take away from these observations? Microorganisms have the potential to be exchanged between peoples and animals, either directly or through the environment. And that reality can be either to the benefit or the detriment of human health, or for that matter, animal health. And managing this complex reality to the benefit of human health requires the deployment of science and policy across this continuum. which has been called the One Health Continuum. And so there are multiple definitions for what One Health means and multiple understanding of how this idea can be exploited. The way that I've described it is very much from the perspective of microbiology, but perhaps more broadly, One Health is the concept that the health of humans, animals, and the environment are interconnected, and a One Health approach consists of multidisciplinary teams working together to solve complex problems to improve health, society, and safeguard natural resources. So how do we take this understanding and consider one particular issue that is of human health concern, antimicrobial resistance? And so what is antimicrobial resistance and why should we worry about it? I'm sure this is something that you're very familiar with, so I won't go into much detail at all in terms of introduction. But the bottom line is this: if you look at this figure here, you'll see a series of red arrows, and at the bottom you'll see a timeline, and each one of these arrows represents the time at which a particular class of antibiotics was introduced to the market. So if you look at the top left-hand corner, you'll see sulfonamides. The tail end of the arrow was the point at which these chemicals were introduced into the market in roughly the mid-1930s. And the pointy end of the arrow is the date at which antibiotic resistance or antimicrobial resistance was first detected for that class of antibiotics. Likewise, penicillin and a number of other classes of antibiotics that have been introduced into medicine. And you'll see that for every class of antibiotics, there's a period of very few years to more years where antimicrobial resistance is detected. And the concern is that as the genes that confer resistance to these different antibiotics accrue within specific pathogens so that they become multi-drug resistant, the options that a physician has available for treating infectious diseases become more and more constrained. And under a catastrophic scenario, mobile genetic elements that carry genes that confer resistance to all the antibiotics that we have available will mean that those infections are not treatable, and that will result in very unfortunate health outcomes. Projections for how big a problem this will become in the future vary, but this is quite a famous one, published in the British O'Neill report 5 years ago. And what you see in this figure here with a series of concentric circles is the current status is globally, there's estimated 700,000 deaths due to antimicrobial resistance versus cancer, which has more. But if you move out over time to 2050, the projection is that if in a business as usual scenario, there would be perhaps 10 million fatalities due to antimicrobial resistant infections, and that will outstrip the projected number of deaths due to cancer. So that is the reality that we may be looking at. And if you think of it in terms of economic cost, it's projected that decline due to productivity, an increase in healthcare costs, morbidity, mortality would perhaps represent $100 trillion US per year. So a very significant sum of money. So when you think of antimicrobial resistance and how organisms acquire genes that confer resistance, and how we can intervene to mitigate that process, there's really three overriding elements. So the first one is the so-called resistome. And so this is the reservoir of antibiotic resistance genes, or genes that can be co-opted to confer antibiotic resistance, that are found in the natural microbial world that would have been selected for or evolve over the last 3 billion years or so in response to the development of a biosynthetic capability for producing antibiotics. So it's this idea that on the planet, there's a reservoir of antibiotic resistance genes that could be available for mobilization into pathogens of human health concern. The second key element is the so-called mobilome. And this is the reservoir of mobile genetic elements, the machinery that has the potential to move antibiotic resistance genes from that resistome ultimately into pathogens of human health concern. So this would include plasmids, transposons, phage, etc. And the third element is selection pressure. This is the idea that in the absence of selection pressure, genes will not be recruited into pathogens. And so really the one element that we can control is anthropogenic pressure. The name of the game is to reduce a reason for pathogens to acquire antibiotic resistance genes. So if you use a drug and you deploy selection pressure through that use, you will lose the efficacy of that particular drug. So when you think about that, and you think about the One Health Continuum and where you can affect change to reduce selection pressure, there's three components to it. So this particular figure here, which has been coined the confusogram, is capturing hotspots, where hotspots for antimicrobial resistance could develop, and the various transmission pathways through which antimicrobial resistance can move across the One Health Continuum. So it looks complicated because it really is, and that is one of the messages that we're trying to convey here, but you can distill it down to three areas of intervention. And so the first area is human medicine. And it's the idea that through judicious use, you can reduce selection pressure for development of antimicrobial resistance in people, whether it's in the hospital setting, the clinical setting, or the community setting. So there's a whole area of research and policy having to do with stewardship in the human realm. The second area is in agriculture, and particularly in terrestrial food animal production systems, where in the West, whether it's in Europe or North America, roughly 3/4 of all antibiotics on a mass basis are used for food animal production. And so there's a whole area of work likewise that has to do with how do you produce food animals while reducing the use of antibiotics. And then the third area is the environment. And it has to do with the waste streams that enter the environment through agriculture or through human wastewater treatment, for example, that go into the environment. And whether in the terrestrial or the aquatic environment, those waste streams are promoting resistance in environmental bacteria that subsequently come back and pose a problem when we are exposed to them. So where does antibiotic resistance develop and what to do about it? In agriculture, we're trying to deploy regulatory tools, adopt alternatives, deploy infection prevention and control, which we would call biosafety, vaccines, other tools, so that we can be better stewards of antibiotic use. In people, same idea, judicious use, prescribing the right drug at the right time, the right dose, not prescribing drugs for non-bacterial diseases, stewardship, deploying good infection control to reduce the likelihood of nosocomial infections in hospital settings. And in the broader environment, effective wastewater treatment, being careful about how we use manures or sewage sludge in fertilizing crop production systems to try to intercept or reduce the fecal-oral route of contamination of people. When you think about this globally, you need to be sensitive to the idea that that there are key differences between high income and low income countries with respect to what drives antimicrobial use in the human settings, in the agricultural settings, and in the environmental realm. So typically in high income countries, there are stringent controls on access to antibiotics. Typically, you'd need to consult with a healthcare worker. You would need to have a prescription. You can't just buy this at the corner store. We have good infrastructure for wastewater, sanitation, and hygiene. So effluents going into the environment tend to be somewhat clean. We have good access to potable water. There are trends that hopefully will drive a reduction in antibiotic use in agriculture. For example, a decline in the desire to consume meat, particularly amongst young people. We have a social license restricting the use of antibiotics. So, for example, the market requesting raised without antibiotics products. That's very much a trend here with chicken, for example, in North America. I would assume it's the case in Europe as well. And we have relatively profitable agriculture. So farmers have some leeway or in terms of how they can change their practices, but still remain profitable. In contrast, in lower income countries, as one would see perhaps in parts of Asia or Africa or South America, for example, there's vastly less effective control on antibiotic access. You don't need typically you would not need a prescription. You could obtain antibiotics through retail outlets. Very little regulation or control on access of antibiotics. Poor education of stakeholders, less of an understanding of the consequences of antibiotic consumption, often very poor infrastructure for wastewater sanitation and hygiene. So in New Delhi, for example, in India, the waste of that megacity, roughly 70% of their citizens would go directly into the environment that they live in. Perhaps only 30% of those citizens, their waste would be treated in any way. There's a marked increase in demand for meat consumption in countries like China, for example, and that is greatly accelerating the use of antibiotics in that country, and therefore the likelihood of resistance being developed in their commercial production systems. And there's a vastly different business model for agriculture. So if in North America or Europe, we can find a vaccine that reduces the need for antibiotics in, say, pig production, but that vaccine represents, although the cost is low, nevertheless, it represents a cost that could not be borne by a farmer in Southeast Asia. It would not be deployed. So as we think through alternative strategies, we need to be sensitive to the realities of what is or isn't possible in different parts of the world. So a little more detail on the agricultural dimension of AMR, again, in this One Health context, and how we can reduce antimicrobial use. So one area of intervention that is quite promising is nutritional strategies. How do we feed animals to keep them healthy so that they don't get ill? And we don't need to use antibiotics either prophylactically or therapeutically. And one area actually being done by a colleague of mine here in Canada is getting very promising results with berry pomace, cranberry, blueberry. So the skins of these fruits, after the juice is squeezed out, those skins contain a variety of chemistry that would seem to have beneficial effects on the composition of the microbiome in these animals, their immune priming, and their resilience to disease. So that's just one example. A second area, which is an area that we work in, has to do with the interface between agriculture and the environment, and where we can intervene to reduce emissions of pharmaceutical residues and antibiotic resistance genes into terrestrial crop production systems, where they can contaminate produce or crops, or where they can move to adjacent water resources and represent a threat of exposure through water consumption. And so what this chain represents is the continuum between the barn, if you will, or the person, if you will, and the environment, and where we can intervene to mitigate transmission and exposure. So let's just look at animal production for a moment. So clearly, the most effective way to do this would be to reduce the consumption of antibiotics. And that would have the advantage of reducing the emission of contaminants through manure, and also reducing the exposure of humans to these contaminants through the food chain. So if we can do this, we have a double benefit there, and that is an area of active research, as I've indicated. When animals excrete, they will excrete antibiotic-resistant bacteria that were selected for in their digestive tracts, and also frequently residues, chemical residues of antibiotics that remain unchanged. And in many production systems, for example, poultry, swine, dairy, the waste from those creatures are stored for some period of time before they are put on a cropland as a source of fertilizer. And so this represents a second area where we can intervene to make that material more benign before it's deployed into the environment through, for example, composting or anaerobic digestion or other means to change the chemical and microbial composition of that material. And finally, we can regulate where and how these materials are applied to reduce the risk of crop contamination. So for example, if you're a farmer and you're producing vegetables, there would be a mandated delay between the period of time where you're applying manure and the time when you can harvest crops for human consumption, so that these contaminants are destroyed in the soil and reduce the risk of transmission to crops. In many jurisdictions, the waste from people are used as a fertilizer, both in Europe and in North America and elsewhere. There's really nothing that we can do to change what drugs people consume, and nor should we plan on doing that. But the waste from people go into wastewater treatment plants that may be more or less effective. The effluents from these plants may go directly into aquatic systems or be used for irrigation water. The solid material from these plants is typically recovered as sewage sludge and used as a fertilizer. And so this represents another area where we can intervene. We can treat the sewage sludge through anaerobic digestion, chemical means, physical means to change its composition. And we can again mandate how these materials are used. For example, mandating a long period of 1 year between application and harvest of crops for human consumption. So this is the way that we would think about where we can intervene in this continuum to mitigate risk. And I like to put this slide up just to indicate that sometimes there are routes of human exposure to agriculture that we may not have thought of that can be problematic. Thinking about this One Health business, and again, contrasting high income with low income, one element is the proximity of people to farms or live animals. And in many settings, you have, in arid parts of the world, you have irrigation of crops with recycled water from human waste, and that has to be managed to reduce exposure of chemicals. In Asian settings, you often have crop production in close proximity, in peri-urban areas, so in close proximity to human settlements, and you have these live animal markets that can expose people to microbiology from a variety of animals. And of course, this has been in the media quite a bit recently because of concerns about where the SARS-CoV-2 core-2 virus may have come from. And this is in contrast with what we'd experience here, for example, in North America, where our farms typically are in rural areas and physically separated from people. So a couple of words about the chemicals that we are putting into the environment, and that we are putting into the animals, and that we are putting into ourselves through food consumption. And the example that I'll give here is simply the variety of chemistry that is carried in sewage sludge that is used as fertilizer. So when people consume antibiotics or other pharmaceuticals and excrete these chemicals, And those chemicals go into wastewater treatment plants. If they are not readily biodegradable, and if they are sorptive or somewhat sorptive to organic matter, they will partition into the sewage sludge that's recovered from the sewage treatment plant. And what you see in this table here is a list of chemicals that are rank ordered according to their concentration in a sample of sewage sludge that we used in some experiments here in London, Ontario. And you'll see here that triclocarban, which is a biocide commonly found in disinfectant soaps, for example, in this particular instance, was found at a concentration of 6 milligrams per kilogram. Ciprofloxacin, the fluoroquinolone antibiotic, again close to 6 milligrams per kilogram, and then a variety of pharmaceuticals going down to diazepam or Valium at a much lower concentration. Those chemicals that are highlighted in yellow are biocides or antibiotics, and you'll see there are a variety of fluoroquinolone antibiotics, tetracycline, macrolide antibiotics that are found in this sewage sludge that we're applying as a fertilizer in agricultural ground. Some of these concentrations are in excess of the minimum inhibitory concentration. And so this has raised the concern that the concentration of these chemicals that we're deploying in the environment, in this instance in crop production systems, are high enough that they may be enriching for resistance in environmental bacteria. And one of the drivers for this concern is the recognition now that concentrations of antibiotics that are significantly below the minimum inhibitory concentration, so the concentrations that will stop growth that are lethal, can select for resistance. It's this idea that even at sub-inhibitory concentrations, what has been coined the minimum selective concentration, organisms that carry an antibiotic resistance gene that confers for resistance to that antibiotic nevertheless have a fitness advantage over an isogenic clone that lacks that antibiotic resistance gene. So there's a fair amount of work that's being done in terrestrial environments and aquatic environments that are being exposed to wastewater effluents now to determine whether the concentrations of these chemicals that we're putting in the environment represent a risk that we need to manage. So now a few words about food safety and food security. So again, getting back to this idea that microorganisms move back and forth throughout this One Health continuum and can be exchanged throughout this One Health continuum, there is a concern about the exchange of microorganisms and wildlife and food animals and how this can threaten food security. And the two examples that I'll give here, one of which is in the news all the time, is African swine fever. And it's this idea that wild pigs or wild boars are reservoirs for the virus that causes this disease. Those wild boars are moving throughout Europe, other parts of the world, that commercial pigs are being exposed to them. The pigs become ill, and this has caused an enormous slaughter of herds in China and other parts of the world that, at least in China, has had a significant impact on the availability of pork in that country. The second example is avian influenza. And it's this idea, again, that wildfowl carry a reservoir for these viruses and can contaminate domestic fowl. And this can result in the destruction of flocks. And certainly it's been a problem, for example, in Southwest France with the duck industry being decimated, for example. So a variety of food safety concern that are in the food supply that we need to manage. These would include enteric pathogens, bacterial pathogens, viral pathogens, parasites. These would include toxins, including fungal toxins, cyanotoxins, and chemicals. And so we need constantly to be vigilant and mitigate fecal contamination along the farm to fork continuum to reduce exposure to microbial pathogens. We need to mitigate fungal contamination of crops to reduce mycotoxin load and mitigate cyanobacterial blooms in areas of filter feeder production, for example, through managing our terrestrial agricultural systems so that we don't emit nutrients into adjacent aquatic resources that promote eutrophication and then blooms. On the chemical front, you need to regulate the use of pesticides. We need to be wary of growing crops and soils that contain elevated levels of metals such as calcium or arsenic, for example. So here's some, just on the food safety front, here's some estimates of burden of foodborne illness. The data is old, but in Canada, which by the way now has a population of 37 million people, lots of people get sick. The costs are elevated. In the United States, I think it's now 330 million. 1 in 6 Americans get ill, very elevated costs. And globally, population of 7 billion people, they estimate that perhaps 2 billion people get ill, and an estimated of 3 million premature deaths due to these infections. Here's another example to drive home this idea that these issues are global in nature, that when we manage food safety issues or AMR, we can't think of it as being regional or national. We need to think of it as being global. And certainly with the global chains of foodstuff transmission, the example here being tuna being harvested here, being processed or canned here, ending up in a household, in this instance in North America, that we need to be worried about what's happening in all these different parts of the world. So very much like climate change, these issues need to be viewed globally. One important area of that's going to be seminal in terms of managing microbial contamination across the food chain and food safety is whole genome sequencing and the deployment of genomics tools for gaining insights into the movement of microorganisms and genes that are associated with antimicrobial resistance or even virulence across the One Health continuum. And so the vision in the future, not too distant future, I think, is that we would have systems that are rapid and they are seamless with respect to doing whole genome sequencing or even metagenomic sequencing on an isolate from foodstuff or DNA extracted from foodstuff to gain insights into the distribution of pathogens and what their attributes might be, and then linking that information with similar or I guess identical surveillance information from the clinical settings and look at the movement of organisms from agriculture to the clinic as rapidly as possible so that interventions can be as real-time and effective as possible. And that these systems should be harmonized internationally, both with respect to how the work is being done, the pipelines, the metadata, the language that's being used to describe these kinds of things. So I think that we can anticipate that both nationally and more broadly, whether it's at the EU scale or the global scale, that these kinds of technologies are going to be extremely useful and beneficial. So looking to the future and food safety, some of the things that we need to think about is globalization of food chains. Need to be more cognizant of food security, need to think about antibiotic resistance more, need to be thinking about the One Health continuum within the context of climate change having significant detrimental impacts in many areas, perhaps thinking more about public-private sector collaboration. And this would certainly be the case in terms of interventions across agricultural commodities, improving the regulatory systems, particularly in middle or lower income countries, capacity building, again, very important thinking about lower income countries. And perhaps all these various facets can best be thought of together, again, within this One Health idea. So just a couple of concluding comments to wrap up here. So one is there's a nexus between food safety and antimicrobial resistance. People are starting to think about foodborne AMR as being a food safety issue. Right now, it's not considered a food safety issue in a real regulatory sense. And the science is not at the point where practically it likely could be. But if one is concerned about human exposure to AMR through consumption of fresh produce or meat or dairy products, how would one get one's mind around managing that? What would a safe exposure be? What would an unsafe exposure be? Whether it's in the amount of antibiotic resistance genes conferring resistance to an antibiotic of human health concern, or would it be the organism that's carrying those genes, or the genetic context, whether they're associated with mobile genetic elements or anchored in a chromosome? What represents more risk? What represents less risk? And what metrics would one use or criteria would one use to make a decision on that? What would be considered an undesirable outcome from exposure to foodborne or environmental AMR? Would it be a health metric like morbidity or mortality or something having to do with healthcare costs? Or would it be events in the microbiome? So, for example, knowledge that exposure to a certain kind of microbiology results in horizontal gene transfer of genes of concern into the human microbiome, where that represents a reservoir that's more problematic in an instance where the person becomes ill and must be treated with an antibiotic. So I think that there's a large gap between exposure assessments, where we can document what a person is being exposed to through food or the environment, but we don't really understand either in the gut microbiome or the consequences of that for human health, what the hazard would be, and therefore a risk assessment. And so there's a fair amount of, I think, insights that merit to be explored here that ultimately could perhaps be used to influence policy, set standards, determine what could best be monitored. Some big challenges regarding AMR. How effective are risk management strategies likely to be? So whether it's in the human setting or the agricultural setting or the environmental setting, if we're deploying risk management strategies, what's the benefit of that in whatever way, whether it's animal health or human health, for example? We still don't really clearly understand if we stop antibiotic use, for example, in agriculture, whether that's going to result in the antibiotic resistance genes that we've selected for to disappear. So there's a question about whether the genie can be put back into the bottle. As I just indicated, we really don't have a risk assessment framework going from exposure to outcome. And within that context, we don't understand dose response, whether it's impact of chemistry on selection for antibiotic resistance gene or evolution and mobility, other phenomena that I haven't talked about today. And this just summarizes what I just said, is a need to understand the relationship between exposure to AMR in whatever setting and an unwanted health consequence, and how we could use that knowledge to influence policy to the benefit of human health. And with that, I would finish. Thank you very much.
Thank you, Professor Topp. Now we have time for some questions and discussion. So please, if someone wants to do some questions, feel free to do it. You can use the chat box, or you can just open the microphone and do your questions. Well, maybe I can do one question to you, Ed, if you don't mind.
Yeah.
My question is related with the soils amendment with manure or sludges. And my question is the risk more on the side of the chemicals that we are supplementing to the soils, or on the bacteria that can be resistant to antibiotics, or both?
Yeah, I think the answer is both. I think in North America and in Europe, on the microbiology front, there are mandated delays that are quite long between application of sewage sludge and the harvest of crops for human consumption. Typically, these would be a year. So I think that that's probably protective for exposure to microbiology if it's managed correctly. But on the chemical front, there is a question about whether the chemicals are at sufficient concentration that they're promoting for resistance in environmental bacteria. And I think that's currently an area of investigation that remains to be answered.
And is feasible to ask to the producers to wait 1 year till they use again the soil? Because I think, for example, in a small country as Portugal, where our fields are not so big and they are continuously being used, I don't imagine that being done, but I don't know.
So my guess is in Portugal, if a farmer is producing vegetables, the farmer would have to wait a year between the use of human sewage sludge and the harvest of vegetables. If the farmer is producing other kinds of crops that would be consumed by animals, for example, that delay would not be so long. Okay. The farmer may also use animal manure without that 1 year constraint as well.
Okay. Well, I don't know if Celia is with us.
Yes, I'm here.
Hello, our expert in antimicrobial resistance. I don't know if you have some questions.
I have. First of all, I would like to thank Ed for the very nice talk. This is one of the good things of the pandemics, that we learned that we may have talks even if we are so far as we are now. It was a great pleasure to have you here, and it is a special occasion for our research center. And I really enjoyed your inspiring talk, because indeed everything is connected, and we see that the environment and the food safety and our health is indeed a continuum. And this has all to do with the research that we do here at CBKF. But I would like to make you a question that is related with something that we feel every day, I think, and everywhere about these pandemics. And is the same when we talk about AMR and other topics. That is that the science always think that have the solutions, and that we can design the solutions, and need and must are keywords that we normally use, because we can think about solutions, but then it is quite difficult to implement that solutions, because as Yvonne was saying, probably we cannot ask a farmer to wait 1 year to produce something in that soil. So how do you think we could somehow bridge between science and policy making and the politicians, and to have a better dialogue than we have nowadays between these two realities? I think this is the big challenge in climate change, in COVID, and also in AMR? What do you think?
So you think I have the answer to that question?
I know you do.
This is the perpetual problem of the human condition. I guess two things. One is if people could embrace this One Health idea, whether it's the politicians, the policymakers, the people who have technical expertise, the industry, that would get people talking to each other more, which would be very, very important. And you mentioned climate change as the other obvious problem where these kinds of issues crop up, and look at how poorly we've managed climate change. In many instances, different stakeholders have motivations that are absolutely confrontational. If you're the fossil fuel industry, you're not open to these kinds of things. So that's going to be a continual challenge, which will require political courage. The second, I guess, more granular answer with respect to resolving some of these problems, and I'll go back to agriculture, is the policy objective is reducing antimicrobial use in agriculture. And farmers are often reticent to do this because the farm is the basis on which their family feeds itself. They don't want to take risk. They've always done the same thing the same way, etc. So the solution there, sorry, the mail is arriving and my dog is protecting the household. Is to provide them with alternatives that they can readily embrace that are effective. So I'll go back to the climate change example that you gave. If the policy objective is to take people off gasoline-powered vehicles, and you go to society and you say, stop using gasoline-powered vehicles now, they will push back because they need to go to work, they need to take their children to the football practice, everything else. But if you say we now have electrical vehicles that are cheaper than gas-powered vehicles and have all these additional benefits, you can reach your policy objective extremely rapidly. So if our objective is to reduce antimicrobial use in agriculture, we need to find vaccines, nutritional alternatives that are as effective, cheaper, easier for the farmer to implement. In the food safety realm, we have extensive systems of codes of hygienic practice, HACCP systems that are protective for food quality across the farm to fork continuum, that are based on mitigating human exposure to human pathogens. We need to validate those systems for whether or not they're effective at mitigating human exposure to antimicrobial resistance. They may very well be, they may not be so effective, they may need to be altered in some way, but I think the strategy is to go into systems that are already being used or that could be used, that are economically viable, that are practical, and validating them.
Show alternatives.
Yeah.
Yeah. We have one more question or two questions from João Paulo Ferreira. I don't know if you want to turn on your microphone or if I should do the question. I will do it.
You can.
Okay.
Yeah. Okay, you can do the question.
Yeah. Okay. So the question is concerning the berries example that you talked about. If the effect that is being observed is because they have an antimicrobial effect, and if in this case the antimicrobial resistance cannot also occur because of that, and if you know other similar examples with other foods or
so the question, if I understand it, would be whether the microbiology in the animal becomes resistant to a component of what's in the extract. And because of that resistance, there's co-resistance to an antibiotic of human health concern. I don't think that that is the case. I would not be too concerned about that. at least in the chicken example that I'm somewhat familiar with, I have a colleague who's very familiar with it. And I would also emphasize that, at least in North America, the broiler chicken production cycle is 36 days. So from the time the chick goes in the barn to the time the chicken ends up as meat is 36 days. So there's very little time for selection of antibiotic resistance to something that's carried in a cranberry extract. I don't know. I don't think the mechanism of that is completely known. There's certainly alterations in the microbiome. There seems to be something to do with the immune system of the chickens, whether it's mediated directly through chemistry in the chicken or chemistry microbiome chicken, I don't know. It's probably a complex process that requires further examination. But that particular piece of work is currently in the hands of our regulatory authorities, who are seeking market access so that farmers can use those berry extracts in the feed for their poultry.
Yeah, that is really curious, because I know that in the Nordic countries, for example, they usually use berry juices or extracts to prevent urinary tract infections.
Yeah.
I know
So,
if results or not, but
yeah.
I know they do it.
Right. So this alternative technology here has the welcome co-benefit that the chickens get fewer urinary tract infections. Or cloacal infections.
Well, anyone has other questions? No. So I have one related with domestic animals. Well, that is something that I was talking with Celia. And my question is, what do you think about the relevance of the domestic animals in the antimicrobial resistance transmission to humans?
Are you talking about companion animals?
Yes, yes.
Okay.
Because if we share the same microbiome,
right.
Probably we also share the same resistome.
Right.
So should we consider them and study a bit more, or they are not major players?
Okay, so two comments. One is the problem here is not dogs, it's cats. Okay, dogs are blameless. Two,
I know you have
it. Yeah. I always protect my canine friends. No, but seriously, I've talked about this with colleagues who are veterinary epidemiologists. And from their perspective, the way to think of a dog or cat that you have in your home is the way to think of your children. Basically, the companion animals that you have in your home are people that you share microbiomes in the same way that you do with your children. And when you send your dog to the vet or cat to the vet to get treated for an infection, it's likely that the antibiotics that are used are human relevant antibiotics. But you'd have the same thing to say about your children. If you have a child that goes with an inner ear infection or whatever infection and is treated with antibiotics, and their microbiomes are under selection pressure, you will be exposed to those as well. So I think maybe the way to think about companion animals is not as a discrete, separate entity, but the way that you would think about household members.
Okay.
I don't know if you would agree with that, but.
I don't know. I'm just starting to think about that, so I don't know.
It's interesting because this comes up all the time, and nobody has a responsibility for companion animals. So, for example, in Canada, as I'm sure in Portugal, we have a Ministry of Agriculture, we have a Ministry of Human Health, we have a Ministry of Environment. We have no Ministry of Companion Animals. So nobody takes responsibility for them.
Thank you. Do we have more questions? I don't have any in the chat box. Someone wants to ask something? No? So, Celia, if you want to say some final words. or ask something. I don't know.
I would like to invite Ed to come to our research center one day and visit us and stay here for some days. Portugal is quite nice, so we have a lovely sun now at this moment. So you are invited to come when the pandemic is finished, maybe in 2 years. Okay.
Thank you so much. I will do that.
Not 2 years. This
I
is
hope
your.
not. I hope not.
To be
I think
safe.
2022.
Yeah.
Yeah, maybe.
1 year. Okay. Okay. Thank you, Ed. I don't know if there are some more questions. If not,
No.
just let me say thank you very much for being with us today, and I hope that one day we will receive your visit in
Thank
person.
you so much.
Thank
Thank
you.
you for the invitation again.